Ladies and gentlemen, on behalf of Invivoscribe, we welcome you to this webinar hosted by AMP, the Association for Molecular Pathology. Our program's title today is Minimal Residual Disease Detection of B lymphoid and plasma cell neoplasm using an NGS-based clonal IgH rearrangement assay. We're very pleased to announce our two presenters. Interestingly enough, we are literally covering the United States from coast to coast today by having a presenter from New York as well as Los Angeles. Our first presenter is Dr. Caleb Ho, medical doctor. Dr. Ho is an assistant attending pathologist of both the hematopathology and diagnostic molecular pathology services at Memorial Sloan Kettering Cancer Center in New York. Dr. Ho received his medical doctor from UCLA David Geffen School of Medicine. After that, he completed his anatomic and clinical pathology residency at Yale School of Medicine, followed by a hematopathology fellowship at the Brigham and Women's Hospital and a molecular genetic pathology fellowship at Memorial Sloan Kettering Cancer Center. He has a particular interest in the validation and implementation of molecular testing as related to hematologic malignancies. Dr. Ho's presentation is titled Minimal Residual Disease Detection Techniques and Residual Disease Quantitation in Lymphoid and Plasma Cell Neoplasms. Our second presenter is Dr. Ryan Schmidt, medical doctor, PhD. Dr. Schmidt is Assistant Director of Clinical Genomics Laboratory in the Center for Personalized Medicine in the Department of Pathology and Laboratory Medicine at Children's Hospital, Los Angeles, and an Assistant Professor of Clinical Pathology at Keck School of Medicine of the University of Southern California. Dr. Schmidt received his PhD in neuroscience from the University of California, Los Angeles, and his MD from UCLA David Geffen School of Medicine. He also completed a molecular genetic pathology fellowship at Harvard Medical School and a clinical pathology residency at Brigham and Women's Hospital. He is focused on advancing patient care, the development, implementation, and interpretation of molecular diagnostic tests. Dr. Schmidt's webinar presentation today is titled Ongoing Development and Validation of a Minimal Residual Disease Assay for B-ALL using Invivoscribe's LymphoTrack test. I am now pleased to turn the time over to our first presenter, Dr. Caleb Ho.
Thank you for the kind introduction. My name is Caleb Ho. I'm from Memorial Sloan Kettering Cancer Center in New York. From the laboratory standpoint, mineral residual disease or MRD detection can be done primarily by two different methods, flow cytometry and NGS-based assay. I have two pictures here showing you the two different methods for MRD detection, but the focus of the talk today will be focusing on NGS-based assay. Just as disclosure, I received honorarium from Invivoscribe, the company that manufactures the LymphoTrack assay described in this talk. Before I get into the details of the test, I just want to provide you with some background. Each immunoglobulin molecule comprises of two identical heavy chains and two identical light chains. The immunoglobulin heavy chain gene, the IgH gene, is located on chromosome 14q and is comprised of different segments known as the V, D, J, and C segments. When the B cell develop, as the B cell develop, it undergoes VDJ segment rearrangement. And this is a process where different segments of the VDJ gene segments are joined together, forming a diverse immune repertoire. Because of the possibility, the enormous possibility of VDJ combination and additional modification to the IgH gene after the rearrangement process, the diversity of the immunoglobulin immune repertoire is estimated to be at least 1 × 10 to the power of 12. This formed the basis of the IgH gene rearrangement studies, where if you see a population of cells with the same gene rearrangement, you can infer that they arise from the same precursor. This is a flow chart. It's a simplified flow chart showing you the workflow for the lymphotract assay. After extracting DNA from the sample, the first step will be amplifying the gene segment of interest using PCR reaction. So the lymphotract assay provides different primer sets for amplification. For 4 primers, you have primers that bind to FR1, FR2, and FR3 regions. And these are the primers that are further optimized from the Biomed2 recommendations. There are additional primer sets that bind to leader region, which is upstream of the FR1 region, and these are the primers that are not described in the Biomed2 recommendation. For reverse primers, there are consensus J primers that bind to the J segment region. After the amplification step and the sequencing step, the next step in the workflow is grouping and merging of the clonal type sequences. The Lymphotrack software can do the step for you where it take all the sequences and merge any sequences that are within 2 base pair of each other. The merging step allow us to take into account any PCR error and sequencing artifact that can be introduced in the process. After the merging step, each clone type sequence is compared against a reference database. You can manually take the sequence and put it into a publicly available database such as IgBlast to get the information, but the Lymphotrack software can also does that for you. So this is how the Lymphotrack data output look like. You get a Excel file with all the clone type sequence organized by descending order of percentage of reads. Information that you can get from here include the length of the sequence in base pairs, the VJ gene segment usage, the percentage of sequencing read corresponding to each clonotype sequence, the mutation rate compared to germline sequence. And this information is particularly helpful for tumor types such as CLL, SLL, and mantle cell lymphoma. The software can also tell you whether the sequence is predicted to be productive or unproductive. After comparing the clonotype sequence to the reference database, the next step would be calling a clone sequence based on the available information. As you may be aware, in order for MRD detection to work, you have to characterize the disease clone, preferably in a sample with high level of tumor content. And after you characterize the clone, then you can search specifically for that sequence in subsequent monitoring sample. So based on the experience from our institution, sequencing hundreds of tumors from different tumor type, we have established a clonal clone criteria that was published recently in the Journal of Molecular Diagnostics. And this is a very complicated table, but the most common scenario that you see will be highlighted here, where you have a sample with over 50,000 sequencing reads, and there's 1 to 2 dominant sequence. In order to call a sequence a clonal sequence, we recommend the sequence to be at least 2.5% of the total number of reads and 10 times the background. And sometimes you can see multiple clones in the sample. This chart here shows you the experience that we had at our institution characterizing the clones from different tumor types, and these are all B lineage neoplasms. From my experience, using a combination of FR1 and leaded primers, you can successfully characterize the majority of tumor types, as you can see here. There are certain tumor types that are hard to characterize and may require additional primers such as FR2 and FR3 primers. And these are usually tumor types with high somatic hypermutation rates, such as follicular lymphoma, plasma cell neoplasm, and diffuse large B-cell lymphoma. After you successfully characterize a disease-associated clonal sequence, then you can search for this sequence in a subsequent monitoring sample. The Invivoscribe Lymphotrack MRD software can does this step for you, and it's pretty simple. You plug in the sequence, as you can see in the screenshot here, and it will search for that sequence in the sample, and it will search for sequence with exact match, as well as with 1 or 2 base pair mismatch from the sequence that you enter. And the search for sequences with 1 to 2 base pair mismatches to account for any PCR error and sequencing artifact that can be introduced during testing. There are two screenshots here. The screenshot on the left shows you how a monitoring sample will look like if the sequence detected, and the one on the right show you how the output will look like if the sequence is not detected. The frequency listed here is the percentage of the clone sequence that was detected as percentage of sequencing read. So next, I just want to show you two examples of tumor types that can particularly benefit from lymphoid tract MRD analysis. This is a case of plasma cell neoplasm that we had at our institution. In the characterization sample, there is extensive involvement of the marrow by a kappa-restricted plasma cell population. And by lymphoid tract, we have a good number of sequencing read, over 690,000 sequencing read, and a clear clonal sequence was detected. The patient was treated and we received a monitoring sample from this patient. By flow cytometry, a minute population of abnormal plasma cell was detected. It was also kappa restricted. If you look at the output from the lymphoid tract data, there's no distinct clonal sequence that you can see here. But since we know the clonal sequence that is associated with the patient's disease, we can specifically search for that sequence in the sample, and the clonal sequence was detected in 0.5% of the sequencing reads. This is another case where the tumor type can is particularly beneficial for lymphoid tract MRD analysis. This is from a patient with BALL, B lymphoblastic leukemia, with an ETV6 RUNX1 translocation. In the characterization sample, we have a good number of sequencing read of over 760,000 sequencing read, and 2 clonal sequences were detected. This corresponds to the results from a capillary electrophoresis-based assay where 2 clonal peaks were also seen. After the patient was treated, we got a monitoring sample where morphologically there's no increase in blasts, and by flow cytometry, there's no abnormal immature B cell detected. FISH study was also negative for the translocation. However, the known disease-associated clonal sequences were detected in about 4% of the sequencing reads. Luckily, because the disease is associated with a ETV6 RUNX1 fusion, we're able to do quantitative RT-PCR, and this particular fusion was detected at a very low level at 0.2% by the RT-PCR assay. So the RT-PCR assay and the lymphoid tract assay provide evidence that there's still a very low level of disease in this patient's marrow. So next, I want to talk about things to pay attention to when you're doing MRD testing. So say if you want to achieve a sensitivity level of 10 to the -6, if you think of it in a very simple way, each cell has about 6.5 picogram of DNA. And if you want to achieve a sensitivity of 10 to the -6, you have to detect one cell out of 1 million cell. And that means that you need at least 6.5 microgram of DNA. But in reality, the detection to achieve a certain confidence level for a certain sensitivity level, there are other factors that involve. So I have some graphs here. These are theoretical graphs and that show you the correlation between DNA input, sequencing depth, and the percentage of confidence in detecting a clone in a particular sensitivity level. So if your target sensitivity level is 10 to -4, you don't need that much DNA input and a relatively low sequencing depth in order to achieve a high confidence. On the other hand, if you want to achieve a sensitivity level of 10 to the -5, you need a much higher DNA input and a high sequencing read depth in order to achieve that confidence level. It's not just the DNA input that can affect your confidence level in detecting a clone. If you run multiple replicates of the monitoring sample, each with a lower DNA input, you can achieve the same level of confidence in detecting a clone. And this is particularly helpful if the clone is present at a very low level. So compare this graph here where if you run multiple replicates, say 3 or 4 replicates, each with 1 microgram of DNA input, you achieve a very high confidence in detecting a clone at a level of 10 to the -5. Compared to if you only run 1 replicate, you need a much higher input to achieve the same confidence level. So at MSKCC, we run at least 2 replicates for MRD detection, and we potentially run more replicates if the sequencing reads for a particular replicate is low. And to call a case MRD positive, we'll have to see the clonal sequence detected in at least 2 replicates. If the clonal sequence is only detected in 1 replicate out of the 2, then we run additional replicates to see whether the results are reproducible. From our experience, we had set a minimum criteria for optimal MRD study, and that's over 200,000 sequencing read per replicate. We'll still report the result if it's under 200,000, but we'll just have a disclaimer saying that the study is suboptimal. And 200,000 sequencing read per replicate for MRD study may seem a little bit low, but you have to account for the fact that in a lot of patients where they're treated, there are really not that many B and plasma cells in the sample. So even if you have a very cellular sample, you may not be able to achieve a very high level of sequencing read. So so we sort of balance all these considerations and set a criteria of over 200,000 sequencing read as a minimum criteria for an optimal MRD study. This is a table giving you an idea of the DNA input and number of replicates and sequencing depth that you need to achieve a particular sensitivity level. And as you can see, if you want to achieve a sensitivity level of 1 × 10⁻⁶, you need a relatively high DNA input with many replicates and a very high sequencing depth in order to achieve that sensitivity level. At MSKCC, we have a validated sensitivity of 1 × 10⁻⁵, and this is the sensitivity that we can routinely achieve. And this is based on consideration of the amount of DNA that we can get from clinical sample. If we want to achieve a even lower sensitivity level, we'll need a much higher DNA input, which may not be feasible in a lot of situations where the clinician have to draw a lot of marrow sample in order to get that amount of DNA. Another consideration is sequencing bandwidth. To achieve a sensitivity level of 10⁻⁵, we are still able to run about 10 to 15 monitoring sample on a MiSeq instrument. But to achieve a much lower sensitivity level of 10⁻⁶, we may only be able to run 1 monitoring sample per sequencing run. And in a high volume cancer center, that may not be feasible. These are some data from our dilution study, and in this study we use cell line DNA. We chose 2 microgram as the input because that's the typical input that we can get from a patient sample relatively easily. So we want to see, using a DNA input that we can routinely get from clinical sample, how low can we detect the clone. So we dilute the cell line DNA to different dilutions, and at the 10⁻⁵ dilution, we can still confidently detect the clone in all the replicates, as you can see here, although the quantitation may fluctuate quite a bit. At a dilution down to 10⁻⁶, we're able to detect the clone at in some of the replicates, but not all the replicates. And so this gives you an idea of why we eventually settle for a validated sensitivity of 10⁻⁵ at our institution because of the DNA input limitation. So based on my experience running MRD samples at our institution, we compared the result between flow cytometry and lymphoid track. And we noticed that for BALL and mature B-cell lymphoma cases, although majority of cases show concordant results between flow cytometry and NGS assay, there's a significant proportion of cases where by lymphoid tract, we're able to detect the clonal sequence associated with the disease, but flow cytometry was negative. In fact, up to 11 to 12% of cases fall into this category. For plasma cell neoplasm, there's a small proportion of cases where there's a discrepant result between the 2 assays, and this could be due to sampling differences between the sample used for flow cytometry and for the NGS assay. These are some additional data showing you MRD detection comparison for plasma cell neoplasm between flow cytometry and lymphoid tract. And there's a high concordance rate, up to 93% of cases show concordant MRD result between flow cytometry and the NGS assay. So finally, for the last part of my talk, I want to focus a little bit more on quantitation of residual disease in the sample. So far, when we talk about quantitation of the diagnostic disease clone in a monitoring sample, all the quantitation is expressed by percentage of sequencing read, which is a number that is not as clinically meaningful to the clinician, and I will explain why. So you started with a marrow sample with a mixture of cells. You have your B cell, hematogones, and plasma cells. The blue cells here represent the other cells in your sample, such as your erythroid cell and myeloid cells, and so on. So during the PCR amplification step using IgH primers, you are only mainly amplifying the DNA from the B cell, hematogones, and plasma cells. The other cells in the samples will have the IgX gene, but they are not rearranged, so they will not be amplified by the PCR reaction. So you take the PCR product and do sequencing on the product. The output that you get, all the clonotype sequences are expressed as percentage of total sequencing read, which correspond to the B cell, mostly the B cell and plasma cells in your sample, rather than the entire marrow sample. So as a way to handle this drawback in quantitation, we've been validating a product called LymphoQuant, which is available from Invivoscribe, which can help give you a way to calculate the disease burden as a percentage of marrow cellularity. So you start out with a DNA input that corresponds to a known number of cells in the sample. So we chose 700 nanogram DNA input because that corresponds to about 100,000 cells in the sample. You add in a known clonal sequence. These are DNA sample that you add into the sample, and we add in an amount that corresponds to about 100 cells equivalents of DNA into the sample. So you mix the sample together, and then you do your PCR amplification and sequencing step. And in the monitoring sample, you will look for the percentage of sequencing reads that correspond to the disease clonal sequence, as well as the percentage of read corresponding to the spike-in sequence that you added to the sample. Since we know that the spike-in sample corresponds to about 100 cells equivalents of DNA, using the information, you can calculate the total number of cells that correspond to the disease clonal sequence using this formula listed here. And once you calculated that the total number of cells for the disease clonal sequence, then you can calculate it as a percentage of marrow cellularity using this formula here, because we know that we start out with a 700 nanogram DNA input, and that corresponds to roughly 100,000 cells. And so you can calculate the disease burden as a percentage of marrow cellularity. So this is meant as an estimate, and you know the numbers that you get can be affected by multiple factors, but this gives you a better idea of the disease burden in the sample compared to using a percentage of sequencing reads for reporting purposes. So we're still in the process of validating using LymphoQuant in our laboratory, but preliminarily from my experience, we have seen better correlation of the calculation using LymphoQuant when compared to the flow cytometry quantitation of disease burden. And we also noticed that the correlation with flow cytometry quantitation seems to be better when the disease burden is low, which we define as less than 10% cellularity. And intuitively, that actually makes sense, because when you have a sample with a high level of disease burden, you have a lot of abnormal B cell and abnormal plasma cells in the sample. They're competing for PCR primer binding and amplification. And that can affect the spike-in product, which is only about 100 cell equivalent of DNA that's added to a sample. So if the spike-in product cannot be amplified well by by the PCR reaction, then that would affect your downstream calculation. Also, from my experience, we think that there might be some benefits from using two different spiking sequences added to the sample. So that way you have two different data points to help you in calculation. That may potentially give you a more accurate calculation of the disease burden in the monitoring sample. So I just want to summarize what I've talked about so far in this talk. As B cell mature over time, they undergo VDJ's rearrangement in the IGX gene, and because of the enormous possibilities of combination in VDJ segments, which are estimated to be at least 1 × 10 to the power of 12, when you see a population of cells with the same VDJ rearrangement, that implies that they originate from the same cell and they're clonally related. The IGX primers available in the lymphoid tract assay include primers set to the leader region, FR1, FR2, and FR3 regions. By NGS method, you get more information on the clonal sequence as compared to capillary or gel electrophoresis-based assay. You get information on the VJ gene usage, the mutation rate of the sequence compared to germline, and whether the sequence productive or not. In order to be confident that you're tracking the right clone in a patient sample, you want to adhere to stringent clonal calling criteria when you're characterizing the patient's initial sample. And we prefer using a sample with high level of tumor burden. And we do have a very stringent criteria for calling a clone, so that way we assure that when we characterize a clone, it corresponds to a patient's disease, not a pseudo clone that may not be disease related. From my experience, across different tumor types, we can successfully characterize the clone in most of the tumor samples using a combination of different primer sets. Once you have characterized the disease-associated clone, you can search for that sequence in subsequent monitoring sample, and this forms the basis of MRD detection. We have a target sensitivity level of 10 to the -5 at our institution, and this is really based on, in practice, the amount of DNA we can get from patient and the sequencing bandwidth available. But theoretically, with the technology of the assay, you can achieve even lower sensitivity. Higher DNA input can increase your confidence in detecting very low level disease. So does running the monitoring sample in multiple replicates. And at our institution, we do at least 2 replicates for MRD detection, and sometimes even more. From my experience for BALL and mature B-cell lymphoma cases, the lymphoid tract assay seems to have better detection sensitivity compared to the 10-color flow cytometry assay at our institution. For plasma cell neoplasm, the detection sensitivity was comparable between flow cytometry and lymphoid tract. And using the technology of LymphoQuant, which is a spike-in sequence mixing in the sample, that helps us calculate the disease burden as percentage of cellularity rather than percentage of sequencing read. Percentage of cellularity, disease as a percentage of cellularity in general would be more clinically meaningful to the clinician than percentage of sequencing read. So I just want to thank everyone that I work with that have helped out in validating this assay at our institution, including members of our molecular pathology lab, as well as members of our hematopathology division, and also a collaborator from the clinical myeloma services. And I also want to thank Invivoscribe for their technical support and for the opportunity for me to give you this talk today. And that's the end of my talk. I'm going to turn over thanks to the next speaker, Ryan Schmidt from Children's Hospital, Los Angeles.
Well, hi, everyone. I'm here. I just need to share my screen. Thank you. I just got the invites for that. Great. Okay, well, thank you so much for the introduction. Thank you to Caleb for sharing all of your wisdom and experience. So hi, everyone. Again, I'm Ryan Schmidt. I'm a molecular pathologist at the Center for Personalized Medicine at Children's Hospital, Los Angeles. And I'm excited to share our early stage development of a minimal residual disease assay for lymphoblastic leukemia using Invivoscribe LymphoTrack. And I have no relevant disclosures. First, I'll start with a little bit of background about Children's Hospital Los Angeles. We're a large center for pediatric lymphoblastic leukemia diagnosis and treatment. We're performing approximately 1,000 flow cytometry-based MRD tests per year. All patients are currently assessed for MRD, by flow cytometry at multiple time points. And we are a Children's Oncology Group COG approved regional reference lab for patients enrolled on COG clinical trials. So given the needs of our institutions, we wanted to stay ahead of the curve for MRD testing and thus considered developing an NGS-based MRD assay. We know the potential for increased sensitivity over flow, as well as the potential for increased prognostic significance of a negative result. And after evaluating several kits, we chose Invivoscribe LymphoTrack. Invivoscribe has long been a leader in molecular clonality testing, and we were drawn to the DNA-based analysis given that gene expression is not required for detection. And removing expression as a variable makes the interpretation a bit more straightforward. The end result then becomes the percent of reads, which can potentially be converted to number of cells using spike-in controls, as Caleb mentioned. And I will also mention the use of the LymphoQuant control later in the talk. And finally, DNA allows for the potential for better sample stability, which is important to maximize the chance of getting a result from archival specimens from patients that might not have been tested at your institution at diagnosis. So Caleb gave you a lot of the background on the LymphoTrack assays. I'll just put this slide here to show you that for our purposes, we'll be looking at both the IGH locus on chromosome 14 as well as the TCR gamma TRG locus on chromosome 7. And what we're trying to do is generate NGS libraries using primers that lie in the V segments as well as the J segments so that we can capture the junctional sequences here. And remember that in the IGH gene, there are 3 conserved areas known as framework 1, framework 2, and framework 3 in the V segments. And for our purposes, we only use framework 3, and that generates a shorter amplicon, but still is able to capture the junctional sequences. So we divide this assay into 2 parts. At the initial diagnosis, we run the IGH framework 3 and the TRG kit, and the goal is to determine the presence of clonality and identify a trackable sequence that can be later tested in the MRD assay. For specimens, we use bone marrow aspirate, peripheral blood, but we also want to be able to test archival specimens such as FFPE clot section and aspirate slides for patients that might not have presented at our institution at diagnosis. Of course, the sequencing depth we need for this application is low. This is at diagnosis, so the patient's going to have 20% blast or greater. So it shouldn't be hard to identify their clonal rearrangements. And for that, we chose a DNA input of 200 nanograms. Let's contrast this to the second assay that's performed using the same kits when the patient comes back and we assess for minimal residual disease. So in this case, we use either the IGH Framework 3 kit and or the TRG kit, depending on what rearrangements the patient has. And the specimen type will be bone marrow aspirate or peripheral blood. And as Caleb mentioned, we want to harness the power of sequencing as many reads as possible, right? We're looking for a needle in a haystack, so we want to build that haystack as large as possible. And you do that by having a lot of DNA input and a very high level of sequencing reads. And what we hope to achieve is to push the theoretical limits of sensitivity down to somewhere between 1 in 10 to the 5th to 10 to the 6th cells. So in our lab, we have several of the Illumina sequencing instruments. And given that the IGH Framework 3 and TRG amplicons are small, they're compatible with the 300 cycle kits. And that gives you a ton of flexibility to use a sequencing kit that works best with your clinical workflow to obtain the number of reads that is appropriate for your application. So you can see that the MiSeq Nano V2 gives you approximately 2 million paired in reads, and you can use different kits to scale up on the MiSeq all the way up to their standard kit for the V2 300 cycle. And you can also use the NextSeq, which we use for many of our clinical NGS gene panel tests. And that has a mid-output and high-output kits that really allow you to access many millions of reads. So at the top, what you're seeing is a schematic showing that in a single sequencing run, there are 22 indices available that you can use for patient samples, as well as 2 indices for kit controls. So that allows you to run in a single run 24 libraries for IGH Framework 3 at the same time as 24 libraries for TRG. So that's a total of 48 libraries, and even when you run that on the MiSeq Nano, you can still get approximately 40,000 reads per library. And that is a great application for identifying the clonal rearrangement upfront at diagnosis. And I'll build on some of the points that Caleb made about the sequencing depth you need for MRD. Again, this is a modeling exercise that's been done by Invivoscribe. On the x-axis, you're seeing the read depth. So this is 1 million reads, this is 2 million reads. And the y-axis shows you the percent confidence of a negative result. Because what we really don't want to do is get a false negative result due to sampling error. We want to provide enough input DNA and enough sequencing reads to provide a negative result with confidence and have a very high sensitivity. And the takeaway here is that to do that, you want to get on the order of millions of reads, so somewhere up here in the top right. And to do that, it's very helpful to have the flexibility to access some of those larger platform sequencers, such as the NextSeq. And I should have mentioned in the previous slide, but these Invivoscribe LymphoTrack libraries, they can be combined on the same run with other testing that might be ongoing at your institution. So it's very easy to batch them in with other things and potentially obtain those millions of sequencing reads. So here's a brief overview of our diagnostic assay procedure. We're using 200 nanograms of input DNA, which we quantify using Promega Quantis. We perform the library prep using the 2 kits I mentioned, TRG and IGH Framework 3. And we perform a library quantification using the Agilent tape station. Note that you can also use a Kappa real-time PCR kit to quantify your library, but in our lab's workflow, we use the tape station for many of our other tests. So our staff feels that this is a quick and easy way to take care of things, and that's what we've been doing. But you can also use the Kappa real-time PCR as well. For the diagnostic assay, we prefer the Illumina MiSeq V2 Nano 300 cycle kit. And for data analysis, we've been using the included Invivoscribe LymphoTrack software. Sorry, I think there's some images that aren't coming through on the on this slide. I apologize for that. But these are images from the Agilent tape station, and what you don't see is that for several example TRG libraries, you have a library fragment size of approximately 300 base pairs, and then when we look at the electrophoretogram on the tape station, we quantify the concentration of the fragments in the 200 to 700 base pair region, and that's what we carry forward for the pooling of the libraries. So a quick note on that pooling. The kit procedure recommends a minimum of 4 nanomolar concentration for your library pool. And there have been some rare cases where one of the libraries is more poorly performing in the amplification. And we've brought the overall concentration of the pool down to try to rescue that library. And when we did that, oftentimes that impacted our sequencing quality. So on the y-axis, this is the percent Q30, so that's a measure of base quality, and the x-axis is showing you the library pooling concentration in nanomolar. So you can see when we stuck with the kit's recommendation of 4 nanomolar concentration for library pooling, we always got high quality sequencing, whereas when we went lower down to 2 nanomolar, it was quite variable, and sometimes that fell below the Illumina's specification for base quality. So the lesson learned for us was that if you have a poorly performing library in terms of its amplification for whatever reason, just go ahead and repeat it, and typically you get a good result the next time, rather than trying to pool all the other libraries at a lower concentration. So in terms of the sequencing reads obtained, I apologize again. I think we're having some trouble displaying some of the figures here. You can see it over on the side that for total reads on the log scale, for both the TRG and IGH Framework 3 assays, using the MiSeq Nano, we got what we expected. We got somewhere between 20 to 50,000 reads, whereas using the micro kit, we got approximately an order of magnitude more reads per library. So one thing we really wanted to do in our feasibility analysis with the Invivoscribe kits was to determine the potential clinical sensitivity of our approach in our patient population. So what I mean by that is how many of our patient samples are going to provide clones that we can detect using these 2 kits that we could potentially track for MRD. So to assess this, we took 69 diagnostic BALL DNA samples that we had in the lab because they underwent gene panel testing at diagnosis. And these represented all the diverse cytogenetic subtypes we see. So we had hyperdiploid, EPV6, RUNX1, BCR-ABL, BCR-ABL-like, E2A, PBX1, MLL rearranged, and IAMP21. So we felt this was a good representation of what we see in practice, and we wanted to know what percentage of these cases had detectable clones. So when we did this, we found that using a definition of a detectable clone as 5% of total reads or greater, that over 97% of our samples had at least one TRG or IGH clone. And when you break that down, 76.8% had at least one TRG clone, and 88.4% had at least one IGH clone. This plot down below shows you the number of clones identified for each sample. So each color represents an individual sample, and you can see that there's a line connecting 0 clones for TRG and 7 clones for IGH Framework 3. This is a bit unusual. You typically do not see 7 clones in a single sample, but at the same time, it's not unusual to see 2 or 3 or sometimes 4. So we were happy with that potential clinical sensitivity, but was this specific? We were wondering if you would also identify clones in unaffected samples. So what we did preliminarily, we've taken 5 unique unaffected peripheral blood samples. These were in our lab for exome sequencing. So these are individuals that do not have hematologic malignancy or a suspected heme diagnosis. And from these samples, we had no clones identified at the greater than 5% read threshold using either of these 2 kits. So let's turn our attention to the MRD strategy. Of course, we want to maximize sensitivity, and to do this, we need to maximize our input DNA as well as our sequencing reads. So kind of the upper bound of what we hope to be achievable is to have a 1 in 500,000 sensitivity. And to get there, we need to assess somewhere around 1.5 million cell equivalents of DNA, so up to 10 micrograms of input DNA, and throw at that approximately 10 million sequencing reads. So the kit procedure suggests up to 2 micrograms of input DNA per library prep reaction. Of course, you can do multiple replicates, as Caleb mentioned, and that 2 micrograms of input DNA is approximately 300,000 cell equivalents. But we didn't have the best success at 2 micrograms. Oftentimes that overwhelmed the reaction, and you got poor amplification. So when we backed off a little bit on the DNA concentration, we got robust library concentrations. So we did a crude titration here. We haven't hit all of the dilution points, but you can see that when you go down to 1 microgram, you get a useful library concentration that we weren't getting at 2 micrograms, and it's not that different from what you see lower down at 400 nanograms or at 200 nanograms, which is what we're using for our diagnostic test. So preliminarily, we're moving forward with 1 microgram of DNA per reaction. Again, we quantify the DNA, and we perform 1 microgram per reaction using the TRG and or the IGH Framework 3 kits, so whatever is appropriate to the patient. We're also adding the lymphotract spike-in that Caleb mentioned. And then this time we're performing the sequencing at higher scale. So we are using the standard MiSeq V2 300 cycle kit, or putting it on the NextSeq, and we typically use the mid output kit for this. And for data analysis, we're currently using the Invivoscribe Lymphotract MRD software, which is included, and you saw a little bit earlier. So we wanted to assess the potential linearity of this method and sensitivity. So we did a serial dilution, and we took the kit positive control for IGH and TCR gamma, and that control is a clonal sequence that is in a background of tonsil DNA, and then we diluted that down into pooled peripheral blood DNA at a dilution of 1 to 100, 1 to 1,000, and 1 to 10,000. And note that the undiluted controls contain clonal sequences at approximately 5% of total reads. So this is a very rough approximate expected frequency. Note that you're diluting a very lymphocyte-rich tissue, the tonsil tissue, down into peripheral blood. But this just goes to show you the order of magnitude that we're working with. We're getting dilution points where you're expecting somewhere on the order of 5 molecules in a million, and 5 in 100,000, and then 5 in 10,000. So we're really going to test the sensitivity of these kits. And in this initial study, we took 1 microgram of input DNA at each dilution, so approximately 150,000 cell equivalents, and we sequenced that with 1.5 million sequencing reads on the Illumina NextSeq. So one thing we noticed was that that expected calculation was a little rough, and the initial dilution point was off of the line of expected versus observed. But really the important thing is that we found that the number of sequencing reads obtained corresponding to the clone, so the observed frequency, was incredibly linear looking over that range of dilutions. So the linear R-squared value was above 0.99 for both TRG and IGH. One thing we noted also is that for the most dilute sample, so the one on the order of 1 in 10 to the 6, we got no sequencing reads for IGH. So that is likely a function of sampling error, right? Because we were looking for something on the order of 1 in a million, but we only threw at it approximately 150,000 cell equivalents of input DNA. So this is an important takeaway when you're doing the MRD studies, that it's critical to provide enough input DNA and enough sequencing reads to not have a false negative result. But overall, we really saw excellent linearity at the dilutions that we feel to be clinically important for us. So lastly, I just wanted to mention the use of the LymphoQuant internal control, which Caleb also mentioned earlier. Again, this is 100 cell equivalents of IGH clonally rearranged DNA, and we had spiked that into each sample. And you can use this equation right here to estimate the number of cell equivalents. So you take the frequency of observing the clone, so the percent region of reads of your clone of interest, divided by the percent reads of the LymphoQuant internal control, and then you just map that to 100 cells. So we're able to successfully use this formula and the internal control spike in to come up with an estimate of the number of cell equivalents that are represented by the KIT positive control clone in each one of our dilutions. So let me summarize what I told you. The Invivoscribe LymphoTrack kits, and in our case, we've been using IGH Framework 3 and TRG, have been great for us. They've been a feasible way to look at lymphoblastic leukemia MRD, and they've been easy to use. They've been flexible. We've shown a 97.1% potential clinical sensitivity among our BALL diagnostic specimens, and the validation is currently ongoing in our lab. So with that, I'd like to recognize a few people, our leadership in our department chair, Alex Judkins, and our center director, Jackie Beagle, as well as different faculty members that have been involved, Gordon Arasa, Jenny G, Mauricio Gorman, as well as Matt Oberle, one of our hematopathologists who's been the driving force behind this project. And lastly, I would like to recognize 2 members of the world's best laboratory staff, Dr. Gigi Ostro and Philip Lai. And I'd like to thank the Association for Molecular Pathology and Invivoscribe for holding this event, and I'd be happy to take any questions. Thank you.
All right, we want to thank both Dr. Ho and Dr. Schmidt for their presentations today. We'd now like to handle some of the questions that have come in from today's listening audience. The first one I'm going to address to Dr. Ho. Dr. Ho, in your early example, number 2 in your presentation, the plasma cells were 0.2% by real-time PCR. And just a second, I got to read the rest of the question. It moved on me. Okay, excuse me. So the plasma cells were 0.2% by real-time PCR, while 4% by LymphoTrack quantitation. What do you think causes the difference?
So the question was probably referring to the BALL case that I was presenting early on, and the difference in the number really goes back to the fact that when you're using LymphoTrack assay or any comparable NGS-based assay, during the PCR amplification step, you amplify mostly DNA from the B and plasma cells, so you're not really amplifying the DNA from all the cells in the sample. Versus when you're doing a quantitative RT-PCR assay, you are looking at the RNA from the entire sample. So the numbers are not going to correspond exactly because you're essentially looking at different things.
Okay, there was a second part to the question also. It says flow cytometry is known to underestimate the population of plasma cells In your experience, does lymphotract quantitation also over or underestimate the plasma cell population?
So we are only beginning to validate LymphoQuant at our lab, and we still need to gain a little more experience. Um, there's really no other good gold standard to quantitate abnormal plasma cell. By morphology, you're not going to be able to estimate the disease burden for plasma cell neoplasm. So all we can really do is see whether there's a linear correlation between flow cytometry and the lymphotract assay, and there's some discussion at our institution, in terms of what are some of the factors that make flow cytometry underestimate plasma cells, and we think there might be some factors that are not necessarily specific to flow cytometry, that will result in other estimation of plasma cell, regardless of whether you use a molecular-based assay or flow cytometry assay. But I'm not going to get into details in this topic. But the short answer is we do see a good linear correlation between flow cytometry and lymphotract quantitation of disease burden for plasma cell neoplasm.
All right, thank you very much. The next question is going to bounce over to Dr. Schmidt. The listener said, excellent and very informative talk. I have two questions. The first one is, the percent cutoff of calling a sample clonal. The Invivoscribe recommended 2.5%. Why do you use 5% instead? Let's go with that first.
Yeah, that's a great question. I think we wanted to be a little more strict for things that might be clinically meaningful. One of our lines of thinking about this was that when you have a patient at diagnosis for leukemia, you're going to have 20% blast or greater, frequently much greater in our hands. And then if you have a monoallelic rearrangement in 20% blast, that could potentially give you 10% of sequencing reads. So 5% is half of that. So we felt that, given that most of our cases have percent blast or involvement of the malignant clones at 60% or higher, that something that is below 5% is probably not going to be clinically meaningful and is actually more likely to be background. But that was just our line of thinking. We don't have any studies or anything else to map that out, to back that up, other than just looking at the distribution of the percentage of reads of background clones and the percentage of reads of clones in patients that we know to have BALL.
So I
all right, excellent. Oh, yeah, go ahead.
Oh, sorry. I also want to add to that, and in calling a clone, besides using the percentage of sequencing read, we also notice that the background also really matter a lot. If you have a very clean background, then you're more confident that the dominant sequence that you're seeing is truly a real clone, with some exceptions. So if you have a sample with a dominant sequence and the background is very low, we feel that we can potentially call a clone at a lower percentage. So that was sort of our thinking when we published that clonality calling recommendation in the article that we published early on this year.
Excellent. All right. In the interest of time, we want to thank everyone in attendance today at this webinar. We also want to once again thank both Dr. Ho and Dr. Schmidt for their fine presentations and their time in preparing for this session. Please note that the session's been recorded and that copies of the recording will be made available via AMP website, and the presentations will also be made available. We appreciate also those who sent questions in, and there were a number of commercial questions. You can reach us at customer service at invivoscribe.com, and we'll do our best to answer those questions also. Again, thank you for your participation. Thank you very much. Bye-bye.